First of all, perhaps I can ask Sarah to come and address us, and unusually, unconventionally, which I have to be very careful about, we might take a few questions, a small number of questions after she's spoken. Sarah, please.
So hello, yes, so my name is Sarah Madden. I'm a second-year PhD student, and I'll talk you through what I'm working on at the moment. As they said, I'm working on new ways to make anti-cancer drugs. So there's been a lot of improvement in recent years of how we can target cancer. So we see now that 50% of people who get cancer will survive 10 years or more. But there is obviously a lot to do with, 350,000 people a year in the UK alone getting cancer. We need to think about how we can target it better. So there's a few common things here with drug resistance can be obviously a problem. Side effects obviously can be great with cancer treatments. And there's also one other I wanted to point out. Some cancers, such as pancreatic cancer, we see very poor survival rates with just 1% of people who get it, unfortunately, living for 10 years or more. So there's something to think about there. So I'll go into one of the things I'm working on is based on this research done on a protein called Omomyc. So Omomyc is a man-made protein and was made as a model to study cancer, basically, so we could understand it better. And what they did was they genetically engineered some mice and then to express this Omomyc. And so as you can see on the left, in the purple blotches are tumours, and just with a 6-day treatment of Omomyc, they see significant tumour regression. And on the right-hand side, I don't know if this works, sort of, we have a graph of mice with sort of lung cancer kind of model. So if you see the control, they kind of all sort of actually die around 36 weeks. With one treatment of Omomyc, there's significant increase in survival time and about 40% survive. But if you have these episodic treatments of Omomyc shown by the blue and the red, you actually are looking at about 100% survival of the mice. And so I was quite amazed by this. If you could imagine if we could see that in humans, it would be amazing. And so this made me think, I wasn't actually working on this project. I was working in the lab with Gerard Evan when I came across this research. And I obviously thought, why aren't we making a drug out of this? So that's kind of what I wanted to do. There are, I think, a few reasons why no one had kind of come up with this before. And that was because Omomyc is an intrinsically disordered protein. So it has problems with stability and solubility. And this kind of has various implications. For example, it might have large-scale manufacture would be a nightmare. And so I was kind of thinking about ways to kind of overcome these problems. And so I said, why don't we make it into something called a staple peptide? So this overcomes these problems and also has advantages in the fact that you can add extra groups, which may, for example, help it to get into cancer cells, the drug. And so to make this, we did some modelling, and also we used our knowledge of cancer biology to kind of take the active part of the Omomyc protein and then put it in here and kind of staple it up to make it nice and stable. And this kind of we have a lot of hope for, because obviously the studies with the Omomyc protein have gone so well in mice. So that kind of gives us a good starting point. Also, these studies showed very few side effects. So obviously that's kind of, we'd hope to kind of mimic that as well. And so that's kind of going forward what we'd like to do. And also this would potentially be able to hit cancers like pancreatic cancer. So I'd also like to talk about some other research I'm doing. So in the first slide, I talked about various problems with our cancer treatment, one of them being that we kind of struggle to target some cancer mechanisms. And one of the reasons we do that is that many of the interfaces we see in the mechanisms are very large, and our small molecule drugs simply can't block these interfaces. So we need to think about larger drugs to do this. And obviously that's why, one of the reasons why antibody drugs have become so big. But these obviously, like many other drugs, can have their issues. Sometimes they take a long time to develop, and you never know how stable or how tightly they're going to bind their targets, and how soluble they're going to be. So I am developing a new alternative called a RP, or repeat protein drug. And so the good thing about this is, so I've made it for another cancer target called KEAP1, and it binds tightly, as you can see on the left, so it's a KD of 22 nanomolar for those who are interested. And on the right-hand side, I show the stability, the melting temperature of about 72 degrees to show it's stable. And what I'm working on now is rolling this out to really standardize the process so we can roll it out to more cancer drugs and make it a very quick method to target various forms of cancer. So yeah, I'd just like to thank all those people I've worked with, especially Trevor for answering all my annoying questions, and my funders on the right, especially CSAR and SCI as well. Thank you very much.
Sarah, don't run away. Thank you. That was great. We've got time for one or two questions from the floor, if you would like. If you would catch my eye, somebody down the front here, please.
What's the mechanism?
Could you wait for the mic?
Sorry.
Thank you.
Sorry, what's the mechanism that makes this molecule work?
So Omomy.
What's the mechanism that makes this molecule work?
So Omomy is thought to be, so it's a MYC inhibitor. When you're looking at how it works exactly, it's not exactly fully known. So we can think about it, MYC exists within a network of proteins. So it's thought to, it can bind to a couple MYC and MACs in that network. So it's thought to, the hypothesis is that it perturbs the network and shifts it to a more repressive state. So you're kind of reducing the kind of cell proliferation tendency. There's also thought about it just basically when it's making complexes of proteins in that network, it can, block the E-box. So it's kind of, it's a E-box blocker as well as perturbing this network. That's kind of where we think, but it's not conclusive, yeah.
Any more quick questions? Sarah, you're off the hook. Thank you very much.
Thank you very much.